So at least my watch is 5 past, so I will start. So hi everyone and welcome to this webinar that we call Benefits of Using Standard NGS and Flexible Analytical Software Workflows in Clinical Applications. So what will happen today is that I will first tell about what Qlucore is and what we do. And then we will get into RNA sequencing and why that is suitable as a basis for molecular classification of tumors. And after that, I will show the workflow for a sample before it is analyzed in QI. And finally, I will demonstrate the tool live. So let's get to it. Oh, yes, and I should mention also that if you have any questions, please state them in the chat and I will try to address them when it's appropriate. So let's go. Qlucore is a software company and our visualization-based bioinformatics software that we call Qlucore Omics Explorer has been of great service to scientists around the world for over 15 years. This is demonstrated by the fact that it is part of well over 1,000 publications in peer-reviewed journals, as well as in use at 3 out of the 5 largest pharma companies. Our global presence is shown in the map down to the right, where the green countries indicate where our software is utilized, and although a bit scattered, I think it qualifies as a global presence. Now, over time, it has become apparent that the information gained from DNA is not enough to understand complex cellular behaviors as generation and preservation of tumors. And building on our knowledge and experience from multi-omics analysis, we looked into the possibility to find more from RNA sequencing data. And we think that RNA sequence analysis is the next step in cancer diagnostics. DNA is and will continue to be immensely important for molecular diagnostics, but there is information lacking in DNA that is available in RNA. Like expression levels and gene fusion events, for instance. But the reason that we think RNA is the next step is that the technology is also readily available. So when it comes to protein, it is not quite there yet, but anyone that can sequence DNA can sequence RNA, and the evidence for the utility of RNA is also building up. And I have listed a few of them here. So Liljebjörn et al. Show that expression profiling and unbiased fusion detection increase the clinical classification coverage with 6 to 7% compared to traditional methods. And this means that more tumors simply are correctly classified and can thus be appropriately treated, which is a good thing. The World Health Organization states in their latest update that expression profiling is the most efficient way to achieve correct classification of tumors, which is also promising. Furthermore, Baranovsky et al. Demonstrate that expression profiling is 5 times more efficient in predicting drug response compared to WHO-guided clinical classification. And this points to that we are on the right track when utilizing RNA sequencing for tumor classification. But how come then that expression profiling is efficient for this? Well, a very simple answer is that RNA contains information that is not available in DNA. And to expand a little bit on that, I have made this table of capabilities of different sequencing techniques. So if we start from the top, we have targeted sequencing that sequence a defined part of the genome, usually a pretty small part, focusing on specific genes. And it detects SNPs and indels in the targeted region. And if designed for it, it also can detect copy number variations of specific genes. It is also possible to detect gene fusions. But it is not very usual to detect chromosomal rearrangements and epigenetic modifications with targeted panels. And as we go down the table, we notice that we go from targeted DNA to whole exome sequencing and to whole genome sequencing, and the capabilities increase. And when we get to whole genome sequencing, it is virtually omnipotent. So it detects more or less anything with the exception of gene expression. But if we look at whole transcriptome sequencing, then it doesn't look very impressive. It detects SNPs and indels, but and gene fusions, of course, but not much more. But of course also gene expression. But RNA is different from DNA, and through gene expression, it is possible to detect the effects of all other anomalies. As an example, it cannot directly detect epigenetic modifications like hypermethylation, but it will most certainly detect the effect in the form of altered expression of a gene that has been methylated. So the conclusion is that expression profiling can detect the effects of any genetic anomaly by providing a representation of the current regulatory state of the cells in the investigated specimens. And as Sun et al. Show, by analyzing over 1,400 tumors, expression profiling is a robust, unbiased, and highly specific method for molecular classification of tumors. And this builds a pretty strong case for utilizing expression profiling for tumor classification. So what about gene fusions? Well, gene fusions are highly clinically relevant. And in the interest of time, I will not talk too much about this, but the slide demonstrates that the number of drugs targeted towards gene fusion events are increasing and is an important part in, for instance, the improvement in treatment of leukemias and lung cancer in the past decades. And it is also an important part of the risk stratification of patients, even if there is no direct or directed drug available for the specific gene fusion in question. So it is a very important part of molecular pathology. So if we summarize the benefits of whole transcriptome sequencing-based expression and gene fusion detection, we end up with the following. Gene expression subtyping is the most efficient method to classify according to the latest guidelines from WHO. It has a shorter turnaround time and is cheaper than whole genome sequencing. It allows clinical annotation of fusions, and it can potentially replace a number of currently used methods that are labor intense or time consuming, and because of that then expensive. And examples of these techniques are FISH, immunohistochemistry, and different kinds of arrays. And combined, I think this builds a pretty strong case for whole transcriptome sequencing. But the question then is, how can it be utilized in pathology and hematology labs? And how can we go from RNA sequencing data to a classification of a tumor? And that's, of course, why we have developed our tools at Qlucore. There are software tools that automatically turns our RNA sequence or your RNA sequencing data into clinically relevant results. And I will come back to how in a little bit. So currently there are two tools. Qlucore Insights, which is a research use only tool that may be used for initial testing and proof of concept of new classifiers, for instance. development of classifiers. It is available already today, so if you would like to take it for a test, just let me know. You can drop me an email or give me a call, and I will arrange for testing that in your own lab. So, and then we have Qlucore Diagnostics that is completely dedicated to diagnostics, and this tool is under development. The project has come a long way, and once it is completed, it will comply with the new IVD regulations. So our target date for the IVDR stamp is February 2025, and as of now, it looks very much like we're gonna hit that. So what can be done then with these tools today? So this is the current situation when it comes to the models that are available for Qlucore Insights and Qlucore Diagnostics. And at the moment, Qlucore Insights, the B-cell precursor acute lymphoblastic leukemia, the acute myeloid leukemia, bladder cancer, and non-small cell lung cancer. And on top of that, there is a breast cancer model under development, and in a very early stage, also a model for cardiovascular disease. So there is a lot available already now, but there is also plenty more to come, both new models and new generations of the existing models. An important point to make here is that even though the models are updated, there is no need to change the wet lab. It will remain the same, and that makes our tools, in lack of a better phrase, future-proof. But the point is that new discoveries will be made and changes in guidelines will happen, but thanks to our broad approach with whole transcriptome sequencing and our ability to adopt our models, very little will have to change in the lab. The same process can be used. The only difference will be in the software, which is relatively easy compared to changing workflows in the lab. And that is what we mean when we state in the title of the webinar that there are benefits with flexible software. One of the benefits, I should say. So how does it work then? So this is a cartoon describing the path of a sample from a patient to a report. First, the sample is retrieved from the patient. The next step is that a lab prepare RNA from the sample and sequence that. The raw data from the sequencer is analyzed bioinformatically to generate files that are fed into Qlucore Insights. And after that, the analysis is started and the classification and presentation of fusions detected in the sample are performed automatically. Sounds easy enough. Now, if we have a little bit of a closer look so that you will get a feel for what's actually going on and what you need to do in able to utilize our software, I have added some more information to the slide, and we can go through it in a little bit more detail. I use the BCP ALL model here as an example, but the procedure is very similar between models. So a sample is collected, in this case then, from bone marrow or blood, and the total RNA is extracted from that. And after quality control of the total RNA, a library prep from Illumina is used to produce the library that will be sequenced then. TrueSeq stranded mRNA or TrueSeq RNA library preparation kit 2 are the ones, or the library kits that has been tested for Qlucore Insights. And the kits contain material to convert mRNA into cDNA, and after that the protocols are very similar to a normal DNA library prep. And that's why I made the remark earlier that anyone that can sequence DNA can also sequence RNA. So once the library has been prepared, it is used for setting up sequencing reactions and then loaded to a sequencer, and the most suitable ones are NextSeq and NovaSeq from Illumina. It is most likely possible to run it also on a MySeq, but we haven't tested that so far. Now, the raw data from the sequencing is then fed into a pipeline in the bioinformatics software, We use STAR for the expression profiling, and STAR Fusion or Fusion Catcher or Ariba for detection of fusions. And the setup of these tools are specified in a document that is available for all Qlucore Insight models. And what we get out from the different software is aligned BAM files from STAR, and fusion files from the 3 fusion callers. And apart from that, we also need a text file with information that makes it possible to identify the sample. And once we have these files, we feed them into Qlucore Insights, push run, and a few minutes later, we will have a result in the form of a report that specifies how the sample is classified and what fusions that has been detected. And that's it. So now let's have a look at the software. So this is a screenshot of it, but I have promised a demo, so let's do that. So I should share my screen, which I think I am now. Make that smaller. So I will just have a look what it is that you see. You can see my screen, good. So I will start Qlucore Insights by pushing the symbol. So this is the starting screen of Qlucore Insights, and I run it on a Mac, but it works on both Mac and PC, and the look is very similar actually. So the first thing to do then is to provide the data, the files that we just discussed. So first of all, we need a text file with some information about the sample, in this case, a patient. Then we need to select what model we're going to use, and we will use the BCP ALL model, acute lymphoblastic leukemia. Then we need a BAM file, and I have it right here. So this is an aligned BAM file. We also need a fusion file, and I have it here. It comes from STAR, and it is a TSV file, but there are other formats from other callers as well. It's possible to run up to 4 callers at one time. And the reason for that is that they have different strengths. So some of them are better at detecting some fusions. So if you want to get as much as possible out of it, you can use them all. Once we have made sure that these are the files that we want, we push run, and then it will take about 1 to 2 minutes to run the sample. And in the meantime, I will have a look if there are any questions. And yes, I can also take this time to mention something about BCP ALL. So that is acute lymphoblastic leukemia, which is a disease that is mainly affecting children. It's a very serious disease, of course, but it has a relatively good prognosis, and in the Western world at least, more than 90% of the children affected survive. But it is important to make sure that you know as much about it molecularly in order to use the correct treatment. And now it is starting to get ready with the calculations. So now it's done. So this is what it looks like after the run has finished. And as you can see, there are 3 major windows. we have the results and visualization window in the upper left here. We have the Gene Fusion workbench to the left down here. And then we have the report preview on the right. And let's start with the results window, and I will make that a little bit larger. In this window, you will find plots that visualize the results from the analysis. And this one is a PCA plot. I will make it a little bit larger. And so PCA stands for Principal Component Analysis Plot, and it essentially clusters samples based on their similarity. So all the different spheres here, or balls, maybe I can say it's easier, are our reference samples that we used to train our classifier. So these come from different patients, and the closer they are together, the more similar they are in respect to gene expression pattern in this case. And as you can see, we have different classes here, and the classes are represented up to the left here. And so there is BCR-able or BCR-able1 like, the DUX4 rearranged, the ETV6 RUNX1 and ETV6 RUNX1 like, the hyperdiploid, KMT2A, and TCF3PBX1. And all of them are represented here then. The red ball here represents the sample that we put in. And it looks by visual inspection that like it belongs to the purple class. And the purple class, if we look to the left, is the ETV6 RUNX1 or ETV6 RUNX1 like class. And that is also indicated for the sample here. Then we can have a look at how certain we are that we have made the correct classification. And we do that by looking at the probability plot. So these are the different classes here on the y-axis, and on the x-axis there is the probability that it has been correctly classified. And as we can see, it looks very probable that it is correctly classified, and it is classified then as the ETV6 RUNX1 or ETV6 RUNX1 like class. There is another plot here, but we will wait a little bit with that. First, we will have a look at the Gene Fusion workbench. We'll make that a little bit larger, like that. So there is a bit more interactivity here in the results window. You can't do a whole lot of things, but here you can do a little bit more. And there are several filtering options up here. And by default, the Mittelmann database is selected. And in case you are not familiar with the Mittelmann, it is a resource that collects information about chromosomal aberrations and translocations that are associated with cancer. So if it is relevant to cancer, it should be in the Mittelmann database. So if I deselect the databases, we can see that all detected fusions pop up here in the list. So we have detected 22 fusions in this specific sample. Then we can do some more filtering. We can look at interchromosomal and intrachromosomal rearrangements. We can also filter depending on how many reads we have over the breakpoints and how many spanning fragments that we have in the sample. And that has to do with, of course, how certain you can be that this is not an artifact. It is also possible to apply a blacklist to this. So if you know that you have false positives that usually come up, you can omit that from the list as well to make it a little bit faster. And you can also, of course, apply to the blacklist if it's something that is not relevant to the type of analysis that you are running. But let's click the databases here. So these are probably the relevant fusions. So if you click directly on these, you can see that they are hyperlinks, and you will get to the Mittelman database, and you will get the information about the different publications about this fusion. But if you double click on the row here instead on that one, like this, and then we go back to the results window and look at the genome browser, we will get a lot more information, or detailed information about the specific fusion. So we will know where the breakpoint is located, in this case of chromosome 12, which gene, ETV6, and also the base sequence and the amino acid sequence. You will also get to know if there is a frame shift, so that you will have a truncated protein, and so on. You will also down here get how many reads that you have covering this part of the fusion. You can also look at the fusions from the other side. Let's see if we can do that. So if you double click again, you will see it from the RONX1 part instead. And you will also see the same information, but from the other side, so to speak. Another convenient, or I should say popular function is that if you go to the NGS tab here, you will down here get the sequence over the breakpoint. And this is very useful because it makes it very easy to design and create primers to, it depends on what you want, for confirmation of the presence of the fusion, or for later monitoring of minimal residual disease, for instance. So that is a popular feature. Now, when we have had a closer look at the fusions and the classification, we can go to the results window, and everything that we have looked at is in fact already here. And if you update over here the preview, you can also see that the changes I made in the PCA plot, for instance, but that is true also for the probability plot, it will display here in the same way as it displayed when I rotated it, in order to see this red dot a little bit easier. So everything that you see in the results window, it will also appear here. Otherwise, the report contains a description of the analysis, of course. Then there is information about the patient or the sample here. There is a summary of the results. So in this case, it has been classified as ETV6 RONX1, and the level of confidence is 100%. And we also found the gene fusions, the ETV6 one. So that sort of confirms the classification. Then we have the PCA plot. And when we scroll down, we get some quality metrics. Make sure that what we put into the model was okay. Then we have the probability plot and PCA plot in a little bit larger. and then a detailed description of the analysis, what samples, what files we put in, and also a complete list of all the fusions detected. So once you have gone through this, you can add a comment. So for instance, Fredrik, think this is correct. Something like that. And we can, we can add it as a probability. And then we add it like that. And then it will end up in conclusions here. So Fredrik think is correct. 100%. And once you are happy with that, you push create, and then you name your report, click save, and there you have it. So that's how we perform an analysis with Qlucore Insights from whole transcriptome data. Okay, there are no questions that I can see at least, so I will leave you with that, and don't hesitate to get in contact with me if you want to try it out, or if you have any questions that you want to discuss outside of a webinar. And so thank you for now, and hope to speak to you soon. Bye.